Here's a quick review of how to do Punnett squares. Punnett squares are basically going to fall into two categories, either the monohybrid cross or the much scarier looking dihybrid cross. The one thing you need to remember about Punnett squares is that the outside represents the potential meiosis events. These are the gametes, so they should only have one copy of every chromosome. And on the inside represents potential fertilization events, or the probability that a certain offspring will be created. So there are very simple rules on how to remember how to set these things up. Usually for a monohybrid cross, you are only going to be talking about one trait, and therefore, in any monohybrid cross, there should only be a single letter above or beside every single box, combining to make two letters on the inside, representing a genotype that would lead to a phenotype. For dihybrid crosses, these are two trait crosses, and so above each box there should be two letters, or beside each box, but those letters should be different. They should represent two different genes. And so if we're crossing two individuals that, heaven forbid, are heterozygous for both traits, that is going to produce four potential gametes. If you FOIL out, you can do the fronts, the outer, the inner, and the lasts. We have combinations. Notice that every single one of these contains two letters, but they are different letters representing different genes. And we can set these up in our Punnett square thusly, starting with the capitals and working our way to the lowercase. Same thing over here. Now, if you do it this way, you're going to see some patterns emerge as you solve this Punnett square. First of all, every one of these individuals is going to be heterozygous at both loci for both traits, just as parents are. This individual is going to be a one-off. These two are going to be the same. This individual is also going to be a one-off, but these two are going to be the same. And you'll find that there's a perfect reflectional symmetry across the entire Punnett grid, arriving back where you've got two that are the same, one that's on their own, two that are the same, and then lastly, this individual would be the only one that is homozygous recessive for both traits. This is going to give you, if you do have to do a full Punnett grid that is a 4 by 4, it's going to give you a very typical genotype ratio of 1, 2, 1, 2, 4, 2, 1, 2, 1. The first one and the last one can be found right here, and the 4 would be this entire row of heterozygotes. The phenotype ratio is going to be 9 that are dominant for both traits, 3 that are dominant for one but not the other, 3 more that are dominant for the opposite, and 1 that is recessive for both traits.